No, sir.
No question. Ruhul and Shafi, any question from my previous lecture?
No, sir.
Then please tell me, Ruhul Amin, two criteria of probiotics. Uh,
So Probiotics has three types of criteria. First of all, the safety, functionality, and technological usability. In case of safety, it should be isolated from the healthy gut of animal from or plant. Or in case of functionality, it should be uh, safe. It should be safe from the any kind of antimicrobial resistant gene.
Good. The first criteria, the uh, probiotics must be living organism and living beneficial organism, microorganism. Anyway, very good. Shafi, can you tell me a few criteria of the prebiotics?
Prebiotics are non-living materials, and it's a fibrous substance, and which is used for especially for probiotics.
Great. And probiotics and prebiotics. If I ask you, how can you compare and contrast probiotics and prebiotics in your exam? You should be prepared well. Probiotics, they are really very, very important topic nowadays, not only for research, but also for the industry, because lots of industries are now uh, investing on this important topic. Do you have any idea whether in Bangladesh are we using any probiotics in the practical field?
Um,
Yes, sir.
One by one, first rule, I mean.
Yes, sir. In our poultry industry, I have seen lots of pharmaceutical company are they are using uh, different types of probiotic, basically lactobacillus, bifidobacterium, and others. And in case of human, also I have used probiotics uh, mm, in my relatives. And in case of agriculture, uh, you are the example for using probiotics in case of rice field to prevent the infection or other diseases.
Great. In case of poultry industry, you think they are now in practice. How about the cost of the probiotics that have been used in the poultry and dairy or livestock?
Actually, sir, they're using like bolus antibiotic form. Cost I cannot say the exact cost of animal, but I can say the exact cost of human. When I have, it's a very highly cost, around one tablet 100 taka. So far, I can remember that I have purchased from the market.
So, if probiotics are available for the animal industry in our country, either poultry or livestock, is it possible to have the sample from the company?
I'm not sure, sir.
If they are selling, then why you do not get? If you can buy from them.
yes, sir.
Oh yes, yes, sir. We can purchase from them and we can assess their quality or what kind of organism they use.
Yes,
The,
in our laboratory, what we can do, we can easily check really the product they are selling contain any beneficial organism in living form. So it would be a nice, practical work for this course. And even if you collect a good number, we can do two things. One, in the culture media, several culture media you can check, and also you can extract DNA from the material, either capsule or powder, whatever else, and we can check the 16S rRNA gene sequencing to see what kind of microorganisms are there really. There are any microorganisms or what kind of microorganisms, because our farmers, many of the farmers who love organic farming, they thought probiotics are alternative to antibiotic, so it is safe. And but the problem is, in case of probiotics, they are sensitive to the environment. So if probiotic microorganisms were not isolated from the our environment, that is organisms that are uh, in our country, that are growing in our country, then sometimes they may not colonize in the guards as expected theoretically, or in the proved in other countries. So this is why it is an interesting subject to study the probiotic microbiome in the country, chicken, uh, because we still in the market, deshi murgi are much popular and expensive than the broiler. So probiotics present in broiler from different broiler industry, and probiotics present in country chicken, scavenging chicken, uh, different varieties, morpho forms. We can check. In Brahmanbaria there is
Yes, Asil, Asil.
Asil Murgi, Murad. So we can see why they are so powerful in fighting. Uh, uh,
Definitely, sir. It should
we
be
can check
interesting.
their, the probiotic microorganism. Now we have a powerful tool by using sequencing, and metagenomic analysis. Nazmul is very powerful in metagenomic analysis. We can do such a project. So it is not so difficult. Just you collect the birds from different areas, including broilers. So you can buy all those things and bring back to the laboratory and design the experiment, and then collect the sample, extract DNA, and do PCR and go for the sequencing. And after sequencing, I'm sure it would be a very good publication, and even you can do the your thesis. This is one idea for Ruhul. Or probiotic microorganisms are also present in the milk. So
Yes, sir.
in case of goat, our goat milk is very uh, uh, how to say, very nutritious, and
Has immunity.
many people believe that they are good for the health than the cow milk. So you may compare two types of milk. One is colostrum, first milk after the birth of the baby. So you collect the colostrum of cow, as well as the black Bengal goat, and then regular milk of cow and black Bengal goat, same animal. And if you do it in a replication, we can make a comparison of the microbiome present in the cows and black Bengal, and that would be a very good publication. You can understand what kind of probiotic microorganisms are there, and are there any unique organisms that may play a role in the quality of milk. This is really interesting. And in case of Sorority, you can also do a similar experiment in case of rhizosphere, because we have lots of plant varieties. Some of them are salinity tolerant. Some of them are drought tolerant. So salinity tolerant variety, if you select three salinity tolerant variety and three salinity susceptible variety and grow in saline environment in Satkira or some other area, and after certain growth, if you collect the root and shoot uh, uh, and analyze the microbiome there, you can find a correlation between the salinity tolerance of the plant uh, and the microbiome present there. Because it is now almost established that not only plant gene but also associated microorganisms are involved in stress tolerance of the plant and other organisms. Even microbiome, uh, uh, they are not only uh, playing their physiological role, but they also trigger the expression of genes involved in salinity tolerance of the host or abiotic stress tolerance of the host, even the biotic stress tolerance of the host. That means microbiome, probiotic microbiome can enhance the immunity of the host. So this area is almost unexplored in our country, as only 2 students are now participating this course. You can do some research, and if funds are necessary, I can offer you the fund. So other point is that Ruhul and Shafi both can start writing review. For example, Shafi can start writing a review on uh, probiotic uh, bacteria or microorganism. Bacteria would be very specific. A probiotic bacteria on enhancing stress tolerance in plants. Shafi, are you writing?
Yes, sir. Enhancing stress
Yes. So
tolerance.
the role of probiotic bacteria on enhancing stress tolerance in plants. There are lots of literatures. Stress tolerance means heat stress tolerance, drought tolerance, salinity tolerance, or tolerance of other heavy metals, toxicity, and others. Even biotic stress tolerance means pathogenic microorganisms. So if you can make a very nice review, then I shall consider it as your assignment. So you will be evaluated for 10 marks.
Okay,
And
sir.
second, you can publish it as a article in a journal or as a book chapter. So this would be the opportunity for you, because 2 students, and I think both students are very brilliant. I should offer you some opportunity, and that would help you to learn a lot about the subject matter,
Yes, sir.
and you can go deep, and I shall make some comments on your draft, and you can proceed on. And I am sure if you are serious enough and if you are passionate, then it could be published uh, you know uh, in a good journal, or in a good book as a chapter. In that case, what you need to know, you need to browse the Google Scholar, PubMed, and other databases to see whether reviews have already been published on this topic or not. If directly this topic has been reviewed recently, then we can change the review title. Otherwise, if it is published, 2 years or 3 years back, it is good for you because you can find lots of data in organized forms. Then you can update the literature. So this is not only for Shafi, similar advice for Ruhul Amin also. Probiotic probiotic bacteria in animal industry or like that. Uh, so you can also write a review. You can limit it probiotic bacteria in poultry industry or dairy industry or like this. Poultry may be better because it is available everywhere. So you can also probiotic bacteria in enhancing stress tolerance in animals. Probiotic bacteria in enhancing stress tolerance in animals. So you can include poultry, dairy, even other, cattle farming or like that. What do you think, Ruhul Amin?
Oh, it's a very nice topics for research. Probiotic bacteria enhancing stress tolerance in animal, and which is actually
Probiotic bacteria in enhancing stress tolerance in uh, uh, you know,
animal,
in animals. Animals
and it will
in animal industry or animal or cultured animal. How can we say? Or enhancing stress tolerance uh, of animals uh, by probiotic bacteria, by the application of probiotic bacteria, then it means you are, culturing the uh, animals, maybe poultry or maybe, the other large animals, goat or others. So you can make a nice story. I guess there are some literatures available in the databases. So you need to find out the literature as I discussed the case of plant probiotics. So animal probiotics and plant probiotics, if we have two compiled, comprehensive review, you shall write a comprehensive review. And both of this work you can use later in your seminar because you have to attend a seminar course. So in your seminar course you can easily submit that paper and present to the audience, that is your seminar course team members, and that would be very nice. And target is by the end of January. So please mention by the end of January, you shall prepare your, the uh, review. And meanwhile, we shall proceed on several other topics. Is it right, the target?
Little bit more, it should be good, sir.
Okay.
Or
By the end of January, that means 31st January, you can,
okay, sir.
and and meanwhile, you know, what you would do, in the meantime, you can show me even if you first prepare the content, the outline of the review. First, it is important. So in that case, what you do, you search the literature and see some other published review, and based on all those informations available, you can make a content like a book content. So title, introduction, abstract, introduction, and then small title to tell the story, uh, uh, from isolation to the industrial application. And obviously, the last caption head should be the challenges and opportunities, and challenges of using a probiotic bacteria. And other point you must, before the that challenges and opportunities, you put one a section, a subsection is, the mechanism of mechanism of action, that is mode of action of probiotic bacteria in case of plant stress tolerance or animal stress tolerance, right?
Yes, sir.
Yes.
These are the outline I am giving you. So you start from the isolation and you end with the opportunities and challenges. And before that, you must tell the mechanism of their action in the host organism. And then a conclusion and future perspectives, you list the all references that you included. So as two students, I can, offer some time when you prepare the content. I can also see and give you some tips for improvement. And second point is that when you prepare a draft, then I shall put some comments to make it, publishable. For example, you need to add some, tables uh, uh, summarizing the data published in different research articles. For example, someone maybe used Bacillus subtilis or Lactobacillus, a particular strain, and they used, for example, in case of rice, or for example, in case of goat or sheep, and then they found some effect. So you can write growth promotion or disease suppression particular, and then put the reference. So if you make a table of several column, and if you see our past reviews, you can find the structure of the table. How can you design a table? So you first need to, in your outline, you need to make the outline of the table, what data you are incorporating. The second point is that if you know some, mechanisms, that is, they produce specific enzyme already been discovered or antibiotics already been discovered from the probiotics. So, or specific gene is expressed. So in that case, you can make another table related to the mechanism. So one table should focus or summarize the effect, uh, and another one should summarize the mechanism. And you can make an illustration, finally, the possible ways how probiotic microorganism exert a beneficial, that is stress tolerance to the applied animals or plants. So based on your knowledge, that means published in the literature, you can summarize very nice illustration. Simply you can draw by using PowerPoint and summarize the story. And some in your summary, maybe some of the points you can mention unknown, and some of the points those already been known, so people can have a good picture. And obviously in your story, you can also focus knowledge gap, what is unknown, what should be known, or like that for further studies. You can give some indications. If you read some of the very good reviews published in high ranked journal, I am sure you can find lots of more ideas to organize your review. So what do you think, Ruhul Amin? Agree?
Yes, sir. It is very interesting,
Okay.
and I think
And Shafi?
Yes, sir. It's a really nice idea.
How about
I'll
the
try it.
deadline? 31st January 2021, is it fine? Ruhul Amin?
sir, if we had
For
So
the first draft, 31st January 2020.
so I think it's enough, but for our purpose we can further expand.
No, a first draft you need to submit by 31st January, and then I shall take a week or so to put my comments, and we shall finally you address the comments and finally finalize the draft as a publishable form in February. Is
Okay,
it
sir.
good idea? Good plan?
Yeah, yes, sir. It's a good plan.
Okay. Thank you so much. And let us start some of the, lecture slides. Yes. So in our previous lecture, we showed that probiotic microorganisms, this is a table showing the probiotic microorganisms used in human nutrition. They are mostly lactobacillus, lots of lactobacillus strains. They are, bifidobacterium, but other lactic acid bacteria also there. Lactobacillus is also called lactic acid bacteria because they can produce lactic acid. Uh, Enterococcus, Lactococcus, Streptococcus, Thermophilus. That means they can tolerate high temperature. Uh, and other microorganisms, Bacillus, Escherichia, Saccharomyces, a fungus also used as a probiotic microorganism for human nutrition. So mechanism of probiotics. Uh, how probiotic microorganisms exert beneficial effects to their host. One is antagonism through the production of antimicrobial substances. Antagonism of what? Antagonism against the pathogenic microorganism because either animal or plant or human harbor lots of diverse microorganisms, and 99.9999% are beneficial. uh, but probiotic microorganisms do not harm the beneficial one, but they harm the pathogenic microorganisms. You can ask how, because through the co-evolutionary process, they are actually symbiotic and they want to see their host healthy. This is why they antagonize the pathogenic microorganisms, the enemy of the host. How? By using antimicrobial substances. They synthesize antimicrobial substances like antibiotics or lytic enzymes, different enzymes which are toxic for the pathogenic microorganisms. This is why probiotic microorganisms are considered as an alternative to the synthetic antibiotics. Synthetic antibiotics, uh, uh, you know, uh, their use is really risky, although we have been using because antibiotic resistance is now a big concern. Through the food residue or environmental residue, antibiotic resistance is developing. Competition with pathogens for adhesion to the epithelium and for nutrients. They not only produce the antimicrobial substances, they also compete with the pathogenic microorganisms for colonizing particular gut or root surface or other areas, because for the that means they compete for the space niche, and also for uptaking the nutrient. For uptaking the nutrient, some probiotic microorganisms, they have the mechanism to outcompete to the pathogenic microorganism to uptake nutrient. For example, they can produce siderophores to uptake the iron or like that. In that case, pathogenic microorganism suffers from the nutrition, and this is another antagonism. And third is modulation of the host. As I mentioned, probiotic microorganisms, they can uh, trigger the uh, immunity related gene expression in the host, and how still all the mechanisms are not known, but it is now well established that colonization of probiotic microorganism in the host, either it is animal or plant, uh, the host immunity is pushed up. That means it induces systemic resistance in the host plant against the pathogen. Inhibition of bacterial toxin production. Inhibition of bacterial toxin production. They can, probiotic microorganism can do quorum quenching, quorum quenching. Quorum sensing, perhaps you have heard about this term. Quorum sensing, Quorum. Quorum. Quorum sensing means bacteria within the community, that means same species, they can talk with each other by using signaling molecule. They are basically N-acyl homoserine lactone. N-acyl homoserine lactone type compounds they use to communicate with each other, whether they will go for faster, reproduction, multiplication of the members, if they find very good niche or good nutrient resources, then they take this type of decision. Quorum quenching means uh, probiotic bacteria can interfere the quorum sensing mechanism of the pathogenic bacteria. That means disturb the quorum sensing system of the pathogenic one, and pathogenic one needs a quorum sensing system for their decision, for their population increase through the reproduction, otherwise to capture the host immune system or like that. So host immune system and pathogenic organisms always fight uh, 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 strongly, and in that case, if probiotic bacteria interfere the quorum sensing of the pathogenic bacteria, then it is beneficial, and pathogenic bacteria lose the competence for infecting the host. So there are lots of new information and new findings are being discovered day by day. The immunomodulatory effect of the intestinal microbiota, including probiotic bacteria, is based on three seemingly contradictory phenomena. One is induction and maintenance of the state of immunological tolerance to environmental antigens, nutritional or inhalatory. Induction and control of immunological reactions against pathogens of bacterial or viral origin. Inhibition of aggressive, inhibition of autoaggressive and allergic reaction. So these are the known and discovered mechanism of immunomodulatory effect of the intestinal microbiota or microflora or probiotic microorganisms. So mechanisms of action of symbiotics and their effects you can see here. Here you can see symbiotics. Symbiotics is the appropriate mixture of probiotic and prebiotic. And probiotic, how they exert, mechanism. Here it is summarized, and prebiotics, how they exert. Here we can see probiotics changes in intestinal microbiota, metabolic effects, they produce different metabolites, and immunomodulation. So three major effect they exert to the host. One is change in intestinal microflora. In case of plant, microflora in the rhizosphere or phyllosphere, or even internal tissues. It can be. So metabolic effects, they produce antimicrobial substances, and they also exert immunomodulatory effect to the host. How harmonization of immune response, because immune response is a complex response in the animal as well as in the plant. Plant has immune response also. So they harmonize so that greater benefit of immunity can be established. Prevent respiratory diseases. In case of animal, prevent respiratory diseases. This is why probiotic bacteria, especially Ruhul, you can search and find they are considered one of the option for controlling the COVID-19. There should be some literature. A decrease in levels of toxins in the gut. Decrease in the levels of toxins in the gut. Toxins means produced by the, for example, salmonella and some other pathogenic organism. Colonization resistance, that means they suppress the colonization of the pathogenic organism, and colonize themselves for that means compete for the space. A positive effect on intestinal microflora and combating diarrhea. That means diarrhea causing intestinal microflora, they are suppressed by the probiotic organism. So support and improve digestive process, improve performances, improve lactose digestion, and deconjugation and secretion of bile salt, and supply nutrients, and lower serum cholesterol. You see lots of positive effects exerted by the probiotic bacteria. But here is the summary. Lots of papers, scientific papers, have been published in each of the beneficial effect, and mechanistic and knowledge are there. So this is a very summary. But what can be done by the application of prebiotics? Changes in intestinal microbiota, because prebiotics are the feed of or food of the intestinal microbiota, so they influence, modulate the diversity and population of the intestinal microflora. Inhibition of carcinogenesis, yes. Some of the organism, for example, Helicobacter pylori, they produce some toxin which are carcinogenic, can cause cancer in the human. So they inhibit the carcinogenesis, immunomodulation, nutrient absorption effects, and pathogen inhibition. There are several others like harmonization of immune response, prevent respiratory disease, because prebiotics are the food of probiotics. So if probiotic gets prebiotics, they also can deliver better effects, so positive effect on the development of beneficial intestinal bacteria, and thus on the host health. Reduced risk of colorectal cancer and other tumors, support of the immune system, and reduce the risk of obesity and metabolic syndrome, protection against the infection, even reduce the constipation and other complication associated with the digestion. You see, reduce the risk of obesity. This is very, very interesting, because obesity is a non-communicable disease and serious problem in many countries, including USA. USA is number one. A large proportion of their population are obese, and many countries, obesity is a big problem. Even in Bangladesh, it is a new problem, because of some class of the citizen. They are very rich and they take too much nutrition, and they are unregulated. As a result, obesity is becoming a problem, and obesity is a cause of many other physiological as well as pathological diseases. Obese person's immunity is compromised. So requirement for potential probiotics, this is probiotic selection criteria: resistance to digestion in upper section of the alimentary tract in case of animals, fermentation by intestinal microbiota, beneficial effect on host health, selective stimulation of growth of probiotics, stability in various food or feed processing conditions. So these are the requirements for potential probiotics for the commercialization. There are several classes of prebiotics. Uh, we discussed about probiotics. Now prebiotics are not digested or only partially digested. Not digested means prebiotics are not the food of the human, for example, or animal. And they are, good for the health of the prebiotics. Not absorbed in the small intestine, poorly fermented by bacteria in the oral cavity, well fermented by seemingly beneficial intestinal bacteria. This is very interesting point. Poorly fermented by potential pathogens in the bowel. Because a pathogen, the prebiotics should not be good for the pathogenic microorganisms. And if it happens, then, the host will suffer from serious diseases. They should be fermented by the beneficial intestinal bacteria, that means the probiotics. So examples of prebiotics: Synbiotics used in human nutrition. There are several, for example, fructooligosaccharides, FOS, probiotics, and then galacto-oligosaccharide, inulin, and then xylo-oligosaccharides, lactitol, lactosucrose, lactulose, soy oligosaccharide, and trans-galacto-oligosaccharides. And Symbiotics, they are the Lactobacillus genus bacteria plus inulin. If you apply these two together, then it is called Synbiotics because they work better. Lactobacillus, Streptococcus, Bifidobacterium genus bacteria with the fructooligosaccharides, and Lactobacillus, Bifidobacterium, Enterococcus genus bacteria with the fructooligosaccharides, and Lactobacillus, Bifidobacterium genus bacteria plus oligofructose, Lactobacillus and Bifidobacterium genus bacteria plus inulin. And these are the commercially important prebiotics and symbiotics used in human nutrition. Even in case of other animal, there are similar kind of prebiotics and symbiotics. So this is all for today, because if I tell you too much, you cannot carry on. And now we can discuss a little bit, and then we shall stop today's talk. So Shafi and Ruhul Amin, and now it is your time. You can ask and discuss some point.
Uh, sir, uh, in the last table, you have mentioned that the combination of prebiotics and probiotics are acting as a symbiotic. So, in case of commercial preparation, so uh, do the capsule are containing both prebiotics and probiotics?
Yes, usually when bacterial formulation, uh, probiotic bacterial formulation, industrially prepared. In that case, they use suitable polymer, which are good for the probiotics, and you can consider them a symbiotic relationship. But symbiotic not only in the formulation, separately, probiotic and prebiotic, combination sometimes suggested to take. So you can take prebiotics separately when you take the probiotic in other form, because the dose of the prebiotic and dose of the probiotic is really important, and in case of capsule and tablet it is difficult to maintain such a dose. Thank you, good question. Shafi, any question?
So in case of some people are, especially for the young child, they have some problem, especially for the lactose problem. Uh, in this case, what can we do for that, especially?
Can you check your microphone, because we cannot hear you clearly, and
Hello.
ask the question again.
Uh, in case of child, newborn child, uh, they are facing lactose problem.
Okay, yeah, some people they suffers from digestion of the lactose. Some people cannot take directly milk; they can take the yogurt or like that. I'm not 100% sure whether probiotic appropriate, dose of probiotic can solve this problem. But it is a nice question. You can check the literature whether probiotic can solve this problem. But it is possibly a genetic problem. And for example, some people in Europe, they have the problem of gluten. Gluten is a protein present in the wheat, and some people they cannot take wheat with gluten, and serious reaction, even deadly reaction, can be happen. In that case, now
Sir,
there
can
is
I add something with you?
research on gluten-free, the wheat, and scientists recently using CRISPR-Cas gene editing, they developed a gluten-free wheat variety. Ruhlamin, do you like to add something?
Yes, sir. As I'm from veterinary background, so from the dairy microbiology course, I have studied the lactose intolerance. So basically, if your first point is right, that in that cases they cannot digest the raw lactose, and that is why we need the fermentation, and you can provide in yogurt, dough, cheese, other format. They can easily digest that, and so many people in our Bangladesh are, they cannot consume milk, and in that cases they are suggested to take the fermented product.
Yeah, fermented product, for example, doi. Doi is basically, uh, you know, uh, contains milk, protein, and all nutrients with lactobacillus, like a symbiotic. In that case, lactose intolerant people can easily take the doi, that is curd, right?
Yes, sir.
And curd is, even I am tolerant to lactose, but if I take uh, curd instead of milk, is better for the nutrition. What do you think, Rulamin?
Yes, sir. It has no different based on the nutritional value. So, from the dairy science, we suggest that to take the fermented product or processed product.
Yes.
Dairy and milk, they have both same, basically very similar amount of nutritional value. That's why it will not, it will benefit the those consumer who are resistant to lactose and are consuming the finished product of milk.
And additionally, lactose tolerant people, taking the card, they are getting the lactobacillus, and
Sir.
lactobacillus has some good effect in the intestine. But anyway, fermentation technology is not new. 5,000 years ago, people started to ferment the food and make the alcohol and many other, yeast for the baking. So fermentation technology hugely been, industrialized in the developed countries, especially in Japan. Japan is champion in fermentation technology. They take, soybean seeds called natto. They have several strain of a few strain of characteristic bacteria to make soybean seed fermentation very sticky and bad smelly nowadays. They remove the bad smell, but incredibly good for the health. So fermented products can easily be digested by all kinds of people. So fermented products obviously good for the non-fermented products. You can get the probiotics in addition.
Yes, sir.
Any question?
No, sir.
Sir, in our daily food, we suggest people to consume vegetables or vegetables. So, vegetables you have mentioned that they have also very less nutrition. So, how they and they feed the uh, probiotics. So, I want to know how they actually help us or benefit our us.
Vegetables, raw vegetables, very good for our health. If you go in any uh, developed country and stay in a five-star hotel, you can see lots of raw vegetables. That is a salad you can find there. Because they contain, vitamins and minerals. Some of the vitamins are, uh, you know, uh, heat stable, but most of them are heat unstable. In that case, you can get, for example, vitamin C or vitamin A, D, E, K. You can find huge amount in the raw vegetables, and most importantly, you can find lots of fibers there. Human intestine, philosophically, if you carefully look at the anatomy of human intestine, it is look like the intestine of grasshopper. I'm not sure when human has started to eat the meat, but human intestine tells us the herb is the best, food for the human intestine, as it looks like the grasshopper intestine. Fresh vegetables offer you minerals as well as the uh, vitamins, lots of fibers in fresh form, as well as they contain lots of microorganisms. So you are getting some plant probiotics, and there is no study so far being conducted the microorganism associated with raw salad. A salad always we take fresh, uh, whether they have directly any beneficial effect to the consumer. But it is interesting, it contains lots of million billions of plant probiotics, and who knows, they may have some effect. I am not sure in intestine, the condition and environment, they are acidic and very high temperature, whether they can survive or not. But they can be an ingredient over there and help our health. And or, as an animal scientist, the uh, vaccine, in case of vaccine, the molecules after digestion of the probiotic bacteria present in the salad may induce the immunity, because their molecules, cell wall and other molecules remain during the digestion process in different form. So they may exert some good effects. This is why fresh fruits and salad every day you need to take some. And salad, there is a little risk also. If you do not clean the salad, sometimes salad can contain, toxic, Escherichia coli or Salmonella,
Induces diarrhea.
even can cause the problem. This is why salad, especially cucumber and onion, they are contaminated quickly with the environmental toxic pathogen. But fresh salad always good if you clean them and if you take, and you can keep you green enough. Thank you.
Sir, in case of
Yeah.
uh, sir, in case of consuming vegetables, so usually we boil it completely. So if we boil it, so I do believe that those beneficial bacteria may destroy or damage. So in that cases, what can we prefer? Totally boiled or half boiled?
Yeah, what you can do in case of, as for example, lady's finger or carrot. What you can do, you can just cut them into small pieces, and you can add them to the boiling water for 1 or 2 minutes, not cook, just, surface, how to say surface. If there is any toxic organisms, you can kill and you can take fresh. Uh, that would be nearly fresh or less cooked vegetables are the best. And the green, vegetables contain the chlorophyll, and xanthophylls and many other pigments, and they are very powerful antioxidant. Vegetables contain lots of phenolic substances, and if you cook them, and they are destroyed, and they are antioxidants, they are incredibly beneficial for the human to neutralize the superoxide. So superoxide generated naturally in our body, they are called reactive oxygen species like hydrogen peroxide, singlet oxygen, or O3. They can damage the DNA and can lead to the cancer. So in that case, if you take regularly vegetables and fruits, you can find lots of phenolic substances, and phenolic substances can neutralize the superoxide. They are powerful antioxidants. But if you cook them and boil them, then most of the phenolic substances, they are, undergo the breakdown, and you cannot get the benefit. Clear?
Yes, sir. So in that case, you will prefer totally boiled or half boiled. I have heard that you consume half boiled vegetables.
Yeah, half boiled or fresh. If you cultivate it
Sure.
fresh, also, for example, cucumber, tomato, carrot, uh, uh, lettuce, it is better to take fresh, and you will get the highest benefit.
Thank you so much, sir.
Sorordi,
Yes, sir.
do you have any question?
No, sir.
Okay, no, sir. Thank you.
Okay.
Then this is all for today, and we shall discuss some more exciting topic in the next class. And you are obviously suggested to browse probiotics, prebiotics, and symbiotics in the net and share your findings through the Facebook group, because science is moving so fast. It is difficult to keep you up to date. Because every 2 years, the global mass of knowledge in any discipline becomes double. So it is better every day to search the literature and find some, exciting discoveries. Thank you so much.